Perfect. So welcome back, everyone, to another Tuesday Lunch with a Scientist. We have a very special guest here, our board president, Andy Giordano. Way back when Meg, Mary Ellen, and myself were starting Headwaters in 2014, Andy was super helpful in the development of our science programming and help us take this idea for how we could improve science education and then share it with tons of people. So a little bit more about Andy here. How are you doing, Andy?
Doing all right, Spencer. How are you?
That's great. So I'm going to embarrass you and do a little introduction, and then I'll get out of the way and let you share your talk. So Andy started off as a biology major at the State University of New York, and then went on to get a master's in zoology from Washington State University. Andy's been a science teacher at Sugar Bowl Academy since 2006. And I have really enjoyed getting to know Andy as a science teacher, and someone who's helped me out with my own teaching. Whenever I get to talk with Andy about science education, I come away with it really excited and with all sorts of new ideas of ways I can share these things I've been working on trying to share with students, and ways to do that more effectively. So as we go through today's talk, you'll be able to comment questions to Andy in the comment section. We'll try and get to some of the timely ones during the talk, but we'll also have a Q&A session at the end where you can ask Andy all sorts of questions about the research he's done and his path in the sciences. The last thing I have before I turn it over to Andy is for any of you teachers watching along, we're going to be posting a set of homework questions about this talk that you can assign for your students with the recording of this video so they can engage with the content a little more deeply. As we love to give students at home exposure to all sorts of people who work in the sciences, it's a great way to deepen that connection. And the re-recording of this video and those homework assignments can be found on our website, as well as on Facebook and YouTube. So with that, I'm going to turn things over to Andy. If you have comments, leave them for Andy. We'll try and get to them as we go and at the Q&A session at the very end. All right, let's see. And
all right, thanks, Spencer, and hi, everybody out there. Thank you for popping in on our shelter-in-place science series, our Lunch with a Scientist, our Tuesday talks. Spencer kind of nailed the intro. So my name's Andy Giordano, as he said, and I serve as the board president for Headwaters Science Institute. After studying evolutionary biology and zoology through college and grad school, I started working at a small private school here in the Tahoe area called Sugar Bowl Academy, and I've been teaching science there for about 14 years. So the title of today's talk is How Studying Science Can Lead to a Fulfilling Life. Now, it's a little grandiose, so just to give you guys a background or some other ways to think about it, largely I'm going to be talking a little bit about my journey as a scientist and how I ended up teaching high school science. I'm also think there are a few other different ways to think about this talk. One is, so now I'm a high school teacher, how do I stay connected to science in my day-to-day life? Or how did the pursuit of science through college set me on my current path? Now, it's my feeling that leading a curious life, a scientifically driven life, offers an awful lot of great opportunity for enrichment and for fulfillment throughout our day-to-day lives. And some of the things that I do outside of teaching to sort of pursue that scientific life include obviously my service as a board member for Headwaters, which gives me a chance to really build a program with Meg and Spencer and combine my experience as a science educator with my background in research so that our organization can help align science education a little bit more closely to what science itself looks like in practice. I also volunteer frequently with other nonprofits, including Trout Unlimited and Cast Hope, in order to work on river restoration, habitat monitoring, and just generally get kids out on the water. I contribute as a partner in the annual Tahoe Basin Watershed Education Summit, which is a local program here founded in conjunction with the Forest Service and SWEP the Sierra Watershed Education Partnerships, to bring students from all over the Tahoe Basin into real field work, collecting hydrology data for the Forest Service. This happens every year in the fall, and it's a great program if you're a local high school student to get involved in. And as a teacher, obviously, at Sugar Bowl Academy, I'm teaching biology, chemistry, AP environmental science, as well as digital photography. And having a background in science also supports my two main personal interests, which are fly fishing and landscape photography. And the skills of observation and prediction that are so crucial in science are also critical for success and enjoyment of both of these pursuits. Now, this life, this crafting of this scientifically based life really started as a kid, growing up, and later on throughout being a student, a scientist, a science educator, and a volunteer. I've always been drawn to watersheds, particularly the kind of small creeks that scatter the Tahoe Basin. I'm a big fan of moving water and all the biomes that it creates, and I really love the sense of serenity that these areas can impart. So as a kid, I was always outside. We lived on a 10-acre wetland outside of Buffalo, New York, and in the summer we'd slog up and down the creeks, catching crayfish and getting dirty and exploring. And we would camp as a family on the Ottawa River in Ontario and in the White Mountains of New Hampshire pretty much every year. Now, these experiences engendered in me multiple passions, primarily a passion for exploration and for the woods and rivers and natural systems that these places create, but also for shared experience. This idea that when we go through life together and influence other people and allow them to influence us, it's just a richer life. So there's a picture from a experiential education trip with 10th grade class back in 2012 on Mount Shasta. Here's another one from one of our orientation backpacking trips where we were performing service for the Sierra Club at the huts around where the academy is on Donner Summit. And so throughout my life, I've sort of carried these joys and these passions with me. They've guided my career choices and my life choices. And I tend to think, and this is something I try to push on my students all the time, that our journey through life is not prescribed for us. There's not one road, one path that we can take to success. In fact, I think the world is full of different roads and hubs where these roads collide, just waiting for us to sort of find them, clear them, and connect them as we make choices and follow pursuits. Now, the reality of that kind of a worldview is that the path isn't always obvious. And oftentimes really worthwhile outcomes or really worthwhile paths that we might choose to take hardly seem worth it until we start poking around. Now, for me, this has always been a key, the poking around, the messing around, exploring something, thinking about it, trying to figure out what to do next. I've never in my life really known what the next choice was for me until I had an experience that sort of crystallized how I should pursue that choice. Now, in high school, for those of you high school students out there, it was AP biology. That was the first time in my life where a course not only captured my interest, but that interest motivated me to challenge myself to a higher level, to perform my very best in an academic setting. And that experience, that success, and the content and skills that I picked up in that course really led me to pursue a degree in evolutionary biology at SUNY Buffalo, where I had a similar experience. I ground through all the prereqs, I ground through the sort of normal undergrad courses you would take to get a degree in biology. And at some point, early in my junior year, I had taken almost all of the available undergrad courses in the department that weren't sort of like assigned to you as a senior. So I made the choice to enroll in a graduate course called Evolutionary Ecology. This course was co-taught by a couple of professors, one of whom was named Mike Webster, who turned out to be a very profound mentor for me throughout my career as a biologist, and also for some of the folks watching, including our executive director, Meg Seifert. And this was the first time I was in a room with less than 20 people. I was the only undergraduate in there, and the content was interesting. And I was motivated to do well in the course. In a lot of ways, I was motivated not to embarrass myself and get thrashed by all the graduate students I was sharing space with, but that was what it took, that path, that push, that interest to really find and embrace the challenge. At the time, I was also an undergraduate teaching assistant for the sort of freshman, sophomore level evolutionary biology course. And one of the things about the Department of Biological Sciences at SUNY Buffalo is that they have a huge focus on pedagogy. And that's almost entirely down to the man in this slide, Clyde Herod, who was really one of my first mentors as a student. And Clyde, or as we called him back then, Kip, is in the Department of Biological Sciences, but he really was a teacher. He was focused on teaching. And most of his recent publications over the past 30 years have been about education, about pedagogy. And he really thought there was a paucity of high grade education in the large public university system. And so he focused on training other professors to do well in their courses, and also training grads and undergrads to teach their peers. And one of his big things that he would never admit to, but which became abundantly clear, was that he felt that undergraduate students who are motivated by course credit as well as interest would outperform the graduate students who were simply assigned that course as something to do in order to get their teaching stipend and continue their research. And that gave me and a lot of other folks the opportunity to teach college courses while still enrolled at the undergraduate level. Now, these pursuits, for me, teaching and learning at a high level within the department, got me an offer to enroll in a graduate summer program in my senior summer called Arctic Microbiology. So we took off to do field work in Nome, Alaska for 3 weeks during that summer. And this was the first really deep and enriching field experience of my life. And it was an opportunity that was provided to me because I showed interest and really pushed myself at this time in my life. So on this course, we were studying population dynamics, so how organisms move around, how long they've been there, how much they interact. Now, these microcrustaceans live in these little kettle ponds like you see in this picture, so they have a pretty hard time getting from one place to another. They can't exactly walk across the tundra to find the other pond. So they're fundamentally living on an island. And as we engage with them, just to give you an idea of what they look like, they're not this colorful by any means, but this is an example of a Daphnia, one of the species that we were looking at. And sorry, I messed up my script a little here. And we were studying their population dynamics. Now, on the surface, what this looked like was a bunch of students traveling around the Bering Peninsula collecting water bugs. But in reality, we were doing a whole lot more than that. We were engaging with a wild and harsh landscape, understanding the system that these organisms lived in. We were also having a complex and authentic and committed field experience. We were experiencing failure and success daily alongside our mentors, many of whom would help determine our present as for the graduate students and our future in terms of the 2 or 3 of us undergrads who are on this trip. And in doing so, we were studying the fascinating glacial history of North America. So even on a research level, we weren't just collecting water bugs. We were analyzing their genetic diversity, their similarities within populations and among populations. And our advisor postulated that due to the glacial refugium that we knew existed at the time, called Beringia, as you can see on this diagram, the white bits are the unglaciated pieces during the last glacial maximum, that greater diversity for these populations would help support this hypothesis that there was this Beringian refugium. So he's looking for evidence of that greater genetic diversity in these populations. To be clear, we're like right in here. And these populations, as compared to the populations that they would have fathered or parented further on down the line. So in order to answer that question, he taught us to use genetics, statistics, and geology in order to test a hypothesis. So a multifaceted approach to what seems like a biological problem. And when the trip was over, he was nice enough to offer me a job, collecting samples and running DNA analyses in his lab. And I spent my senior year sort of transitioning from the standard teenager college job of lifeguarding towards being a lab tech and helping and doing my best to help understand how these dynamics worked and how to collect all this data. Now, that experience was extremely valuable. And if we're honest about it, really the field experience was enough to convince me that I should pursue a graduate degree in zoology. That combined with a little bit of know-how in the lab put me in a really good position to do so. So I had poked around, I had an experience, and I was hooked on this idea. So I headed off to Washington State University to study population dynamics and phenotypic plasticity. I was never really an organism guy. I was more of a system guy. I didn't really care what the animal was or the plant was that I was studying, but in this case it was long-toed salamanders. And as it turned out, in order to pay for that, I ended up being a teaching assistant for 3.5 years there as well. Now, the system that we're dealing with is long-toed salamanders, Ambystoma macrodactylum. This was the lab I was a part of, the Storfer lab at Washington State University. They're now doing a lot of really excellent work on host pathogen dynamics, on understanding species range limits, and on landscape genetics and genomics, similar to the work they've been doing the whole time, but really profound work these days. Here's our salamander, our target species. And what's fascinating about these guys is they live pretty much everywhere in the Cascade and Intermountain West, which means that they can live anywhere, because you don't see any empty counties in here, from sea level all the way up to approaching 10,000 feet. Now, that's a huge amount of variation in environmental pressure that these organisms are going to be facing. And what's more is that we had access via a really generous and supportive graduate student who was at the time at University of Montana named Chris Funk. He had a fully detailed backcountry location data for all of these organisms throughout the Cabinet Mountain Range in western Montana. So to orient ourselves, the Idaho border is just off the slide here. We're really only about 20 miles from Idaho. And this mountain range is extremely remote. At the time, it was the most remote mountain range or the most remote river drainage in the lower 48 states. So when it comes down to it, I followed my love for natural systems and completely stumbled into a master's project that gave me license to professionally explore wild places. So I'm sitting here at the time, 2002, and I thought I would pursue a PhD in biology, become a professor, and remain in college more or less forever. So I bought a digital camera to document my field sites and share this world with my friends. Here's a low elevation field site, slightly higher elevation field site with a moose visitor, a couple other spots. But the reality was bringing that digital camera into my world really unintentionally added the passion for photography into my life. So in pursuit of this degree, I was raising amphibians from eggs to adults, trying to figure out how plastic they were in their larval periods of different temperatures. So I'll explain that in a second. We knew that amphibians were highly plastic and how long they take to metamorphose. So that amount of time that they're a larva, between when the egg hatches and when they grow those back legs and leave the water full time. Long-toed salamanders at sea level can go from an egg through that larval phase to metamorphosis in under a month. We suspected that the high altitude populations were able to take much longer. In fact, we thought they could overwinter, which would mean that it would take almost 14 or 15 months for them to go from egg hatching until they were an adult. What we didn't know was how much genetic control there was over this trait, or how closely related salamanders from different populations were. So let's see what's going on here. We can see this is sort of what an egg ball looks like as these eggs are starting to look like larvae and get ready to hatch. Here's a series of emerging larvae starting to metamorphose. You can see the fully metamorphosed adults over here, and then sort of the stages as we step back, more larval-like as we move across the cup. And then, as you can see in this picture, this is obviously one picture, so it's a little anecdotal, but there's plenty of data to support it. We can see the two size classes of high altitude larvae. One, which would have hatched the previous year and spent the winter as a larva, waiting for the right conditions to metamorphose, and then one that was the young of this particular year that still had another 12 months to go before it was ready to metamorphose. So in order to collect this data, it was basically aggressively moving between Washington and Montana to collect as many samples as I could. And when I was home in Washington, I would spend the mornings in the walk-in cooler, sort of like a large walk-in cooler that you might find in the back of the kitchen, changing water for salamanders, feeding and monitoring them. So we're talking about hundreds of salamanders, measuring them every time for their growth rate and watching for signs of metamorphosis. In the afternoons, I'd be teaching or I'd be working in the lab generating and analyzing DNA. So fundamentally, what I was interested in was the role that environment plays in gene expression. That was my question. I thought it was systematic thinking. I thought it was fascinating. The big, complex, and fun ideas. The field work is outstanding. The lab work was technical, but really rewarding. And the aquaculture, growing those salamanders in the walk-in cooler, was brutal. It was horrible. It was the only part I didn't enjoy. And the amount of time it took to establish these field seasons and to get this question underway and from start to finish was about 3 years. So over 3 years, we came to some conclusions. We concluded, in no surprise to anybody, that the salamanders moved around a teeny bit between populations within drainages and not very much between those drainages. And the further they were away from one another, the longer they would be separated. So that's, a lot of work to find out what you might have guessed to begin with. We also determined that salamanders under constant conditions metamorphosed within a few months of each other. So if we held them, if we raised them at the same temperature, the low altitude fast metamorphosing salamanders would take only a little bit less time than the high altitude long overwintering salamanders, which tells us that there were genetic constraints because there was a difference between them. But those genetic constraints, while locally adapted, amphibians fundamentally just end up doing what the environment tells them to do. So if they're fed well, they'll hang out in cold water as long as you want. If you don't feed them so well, or if that water starts to dry up, then they're going to start to metamorphose. So these 3 years gave us those conclusions. And they also included thousands of miles traveling between eastern Washington and western Montana, thousands of hours logged in the lab, many hundreds of miles walked in the field, several hundred hours of teaching, and one blown up walk-in cooler, along with all the samples inside of it and the statistical relevance that they would have provided to my thesis. In the end, what I learned wasn't so much about the salamanders, but I learned a number of really critical skills for a scientist and for a teacher. One was how to develop experiments to test hypotheses, how to deal with criticism and failure, how to design a field study from the ground up, how to acquire understanding of ideas that I found exceptionally challenging, and maybe most importantly, how to communicate complex ideas to peers, mentors, and students. Now in 2002, you'll remember when I started my master's, I thought I had a track. I thought I was on one road with a distinct outcome. In fact, for most students entering as PhD students or as master's students in biology, that is the track that they want to pursue. When I started my graduate degree, biology, being a biologist was rated among the highest degree of job satisfaction in the US. But by 2005, anecdotally watching our friends trying to get jobs as they finish their PhDs, and also just from a data perspective, as you can see in this figure, only about 8% of students who entered as a PhD student were going to end up being a tenure track professor. Now, that's not meant to discourage anybody who thinks that's the track for them. Hard work, thoughtful scientific patterns of living and a pursuit with commitment to a unique idea will certainly give you that opportunity. But for me, it was pretty clearly time to consider another place my degree could take me. So as I was wrapping up my thesis, I got a call from a friend of mine who's coincidentally the husband of our executive director at Headwaters, Ambrose Toscano. He worked and still works at a small private school in Truckee, California, just north of Lake Tahoe. And they were looking for a high school science teacher. And I applied, and 3 months later, I was employed to teach science and work in the dorm at Sugar Bowl Academy. And luckily for me, this turned out to be a place where I could embrace all of my passions, both personally and professionally. That passion for shared experience, my personal passions for photography, really connecting students with scientific patterns of thought. Sugar Bowl is a ski academy, and we are home to interested and interesting students. We've grown into a great facility. We're on a great mountain. We have a dynamic and driven faculty, and we're smack dab on top of Donner Summit in the Tahoe Sierra. It's a rugged place that really breeds excellence in the face of shared experience. And for 14 years, I have called it home. So here at Sugar Bowl in Truckee, teaching science, I can push students to pursue experiences and seek out a fulfilled life. We believe that immersive education provides a real key to finding passion and thereby fulfillment. So our programs include daily challenge, like designing and implementing experiments in the field or in the classroom. And they also include deep, thoughtful experiences like our senior project program or our experiential education program, where students learn about topics for an entire week off campus from experts out in the field, really chasing down the bones of how systems work. Through these programs and with these underlying philosophies, nowadays I'm focused on supporting students to pursue any scientific future which they happen to be passionate about. So thanks for tuning in, guys. I'm excited to see if there are any questions about the research or about the trajectory, whatever you've got for me.
Sorry. That's great, Andy. Thank you so much for sharing that with us. We really appreciate it. So the first thing that I have is a question for you. Right now is a really tough time for education, both for students and for teachers. And so, given how you talked a fair amount about as a student finding a fulfilling or engaging way to connect or do sciences, I was wondering if you had any tips or suggestions for any of the students out here watching and how they might be able to find a really engaging system or way to engage with science a little bit more deeper than they're doing with all the challenges right now.
Yeah, so I would actually first give some advice for any teachers who are watching, which is that now is really a time for us to engage in a couple of different skills that we often preach to our students. One of which is to embrace failure. Allow yourself to take risks in your educational patterns and your curriculum development. Push the limits of what you can achieve through these remote platforms, whether that's trying to transition labs and activities so they can work at home with household materials, or engaging students in the environment that's around them, available to them during shelter in place. For students, the reality is that you have a lot of tools at your fingertips. The first is the capacity to communicate and show engagement and show interest in the concepts that you're really passionate about. Seeking out your instructor or seeking out other capacities like Headwaters Science Institute for online content that can connect you to the outside if that's your passion, or can connect you to current research and household experiments if chemistry is your passion, or medical microbiology or physics or whatever it is. There are a lot of things we can really achieve pretty simply if we just dig a little bit and ask. The most important skill that any of us as teachers or students have right now is communication, that asking for help, asking for enriching material, whether we're asking the internet or asking our actual instructor.
Well, that's great. If you're curious about something and want to know more, ask your teacher. I'm sure they'd be excited to hear that. Great. So my next question comes from, I believe, one of Ambrose's kids. How does one catch a salamander? They can be fast, they can be cryptic and good at hiding. I don't know if you have any.
Well, full disclosure, it's been like 12 years since I've caught one, but I'm perfectly happy to share some tips. One is having your parents help you to identify whether or not you're in the right range, the right location for a salamander in the right habitat. If we're walking around like in a green grassy field, we probably should be looking for other organisms. But if we're in sort of the right habitat, damp, swamp-like areas or moving streams or ponds, we're going to be a lot more likely to catch amphibians. Amphibians are a lot easier to catch when they're larvae, so they're, tadpoles basically, for the primary reason that they exist at much higher densities when they're younger. So if you're going down to, say, Van Norden Meadow with a net, you can kind of drift that net through the grass and catch a whole bunch of toad tadpoles. As far as salamanders, I think you might want to maybe not look so hard in the Tahoe Sierra for salamander larvae. They're here, but they're a lot more cryptic than the western toads that we have so many of. For adults or recently metamorphosed youth, they're going to like dark, damp places. So a lot of flipping over rocks, a lot of flipping over logs. Whenever we're going exploring like that, let's try to do our best to put those items back where they were if we are unable to find what we were looking for, and just keep going with a lot of patience. Those are kind of the skills we need to catch a salamander.
Sounds great. You have to almost think like a salamander for a little bit.
Yeah.
That's great. Our next question from Anna Wettermark is, what are some of the interesting scientific ideas or passion that kids pursue at Sugar Bowl? What are some of the really cool science projects that you get to help students pursue during your teaching?
Yeah, so as far as student-driven work, the most frequent ones that we come across are in the senior project program, where students are asking ideas in the scientific realm and pursuing them through research over the course of the entire year. So we're getting we've had a number of students who are interested in medicine who engage in medically related projects, asking medically related questions. Some of them, Lauren Dunn a couple of years ago, was studying how we can grow organs in scaffolding, like rebuild organs for organ transplant. We've had a number of students look at different approaches to treating cancer, whether that's sort of the viral approaches that we talk a lot about today, or moving back a few years, some of those other trajectories that students get really excited about. Over the past 7 or 8 years, a number of our students increasingly are going on to pursue science in college, whether that's environmental sciences, biological sciences, medicine, microbiology. We're seeing an awful lot of students take that route. On the ground level, like in our day-to-day classrooms, most of the science work that we're doing is generally prescribed for them by the content that we're trying to get across. But we do see scientifically focused experiential education trips too, primarily studying the regional watershed in the Tahoe Basin, sort of the fish that live there, the ecosystem, and how we can support that ecosystem. That's a big part of that fall Tahoe Basin watershed project that I discussed earlier as well, which is a really good program for connecting students and starting to get them to understand their local system so that they can then apply that knowledge to come up with their own questions about it or pursue other systems.
Well, that's great. Anytime we can get students to do those is awesome. And it's always neat to see what students end up choosing when they're given kind of an open book to go for. I have a question about the salamander system that you were studying. Mm-hmm. So I was wondering kind of what types of predators might be eating the salamanders in the areas that you were studying.
Yeah, so in these pristine mountain habitats, there's a number of things to look at, but their primary predators that are endemic to the area tend to be birds and snakes. There's not a lot of fish native to these high altitude populations. However, sort of the introduction and spread of trout throughout high alpine ponds has had a huge impact on population densities of all amphibians, not just long-toed salamanders. So in Montana, I would typically know that if I was seeing brook trout in in a breeding location, that wouldn't be a breeding location for very much longer. And we see that throughout the Tahoe Sierra with mountain yellow-legged frogs and other amphibians that live in these pristine habitats.
Okay, neat. The next question that we have for you from someone maybe close to your home is your daughter wants to know how fast is a salamander, and maybe if we could compare that to something like a frog or other things.
Yeah, sure. Yeah, she's sitting like 10 feet away from me, so she'll hear this in real time. Salamander larvae are pretty quick, just like a tadpole is pretty quick. So when you're catching them in the water, they're about the same speed as the frogs that are the same age. However, salamander adults are really pretty lethargic. They don't have that jumping capacity that we see in frogs. They're unlike, if you see one, you're quite likely to catch it. You just sort of reach down and pick one up. They're not that quick.
Cool. Well, how does the adult salamander, if it's so slow, avoid some of these predators? What strategies does that use?
Yeah, I mean, they utilize a lot of the strategies that we see throughout nature, whether it's cryptic coloring so that they're a little bit harder to see, trying to match and blend into that environment. They also are generally cryptic in their behavior. So they spend a lot of time as adults underground, under logs, under rocks, sort of living a pretty isolated lifestyle. Salamanders, unlike frogs, don't call to find mates. So there's no point in their life history where they're sort of drawing attention to themselves in the system in order to find their mate. Instead, they're pretty philopatric. They go back to the pond where they were born. They swim around and they mate in the pond. And that's the only time in their life history as adults where they're really exposed to the sort of predation.
Cool. That's neat. And then my last question for you is, I know you love getting out and looking at small streams right now. And I was wondering, right now it's spring, things are starting to melt out with snow and things are becoming a little more biologically active.
Mm-hmm.
You're going out this week or next week or whatever to go outside. What types of things are you looking for or excited to potentially see outside?
Yeah, so number one, in the spring, especially in an alpine environment, this isn't scientific, it's just common sense, is I'm looking to see what kind of flows are going on in the streams and rivers to make sure that I'm approaching them safely and considering sort of what the risk-reward ratio is for getting up close and personal. For me in the spring, a lot of what I'm doing is fishing. So in order to fly fish successfully in systems like this, you start paying attention to what kind of bugs are going on. You start looking for spawning trout to see if there's a lot of eggs. If you go down, say, to the Little Truckee, you can watch the trout spawn along the shores. They're fairly well signed. The reds, or the sort of like sand flats where the trout spawned, are really well signed on that stretch of the river, so to keep anglers from walking through the spawning habitat. On the upper Little Truckee along 89 between Tahoe City and Truckee is another good place to go and observe wild trout spawning. During this time where the trout are pretty vicarious is a good time for birding as well. We can see the large raptors starting to come out and be more active and picking off trout that are behaving a little bit more obviously than they normally would be. Those are the things I'm paying attention to.
Oh, well, that's great. And then we actually just had one last question from Ambrose. I think this one's from Griffin. If we have any ideas for science projects where students could combine photography and data collection.
Yeah, I mean, I'm sure if I thought about it, I could come up with quite a few. But right off the top of my head, I would think any sort of work you guys are doing with the bird feeder projects that Headwaters has set up over the past couple of weeks is a really great opportunity. Not only for sort of presence and absence and usage rate of that bird feeder, but also just to catalog and collect what kind of birds are coming in. Photography is obviously a really useful tool for scientific data. The role it plays in sort of wildlife monitoring is often using remote traps. So setting out a camera in your backyard. They're not free, but they're not super expensive, where you can put that camera trap against a tree in your backyard and just see who's walking through. Since most of the interesting animals are going to be walking through at times when you're not paying attention.
Yeah.
Other ideas could be comparing. It could be interesting to be taking pictures of. Oh, my phone is ringing. That's okay. Other ideas could be around here, looking at like the height of the lichen versus the snowpack. Like it's pretty fun to take pictures of, so you can growing at the green, the green mossy looking stuff that's growing on the side of the trees.
Yeah.
It's interesting, depending on whether you want to go longitudinal and take the same kind of picture every spring to see if it's moving up or down with the snowpack, or you just want to compare like the snow depth in that year to what might be the maximum snow depth, the point at which that lichen starts to trail off. Yeah, I can, what I'll do is I'll think about this for the next week or so and put together a blog post for Headwaters. You guys can tune into it next week. It's a really good question.
Awesome. Well, thank you so much, Andy. We really appreciate you taking the time to share the relationship that you have with science. It's great to hear you so thoughtfully describe that to us, and we appreciate you taking the time and all the help you do with it for us.
Yeah, thanks so much, Spencer, and thanks, folks, for tuning in.
Cool. All right. Take care, everyone, and we'll see you next week.